Good evening, everyone. My name's Amy Lockman. I'm chair of the Australasian Human Microbiome Research Network. And I'm delighted to welcome you tonight to our inaugural network event. Now, just before we get into it, in a classic example of a Zoom glitch, the speakers are actually appearing under my name, but rest assured, I will not be speaking the whole time. We have many more qualified people to speak to you about these topics tonight. Now, the goal of the network is to bring together the Australasian microbiome community. And what better way to do this than to come together to showcase some local talent and discuss the gnarly issues in microbiome data analysis. So that's how we'll be kicking off tonight. So formally, the network serves to advance impactful microbiome research for the benefit of human health and disease prevention. Our mission is to build a community for the sharing of expertise and resources amongst Australasian researchers to enhance the quality of microbiome science and research for human health. We have a working group of 12 committed members who meet monthly to bring you the offerings of the network. And we're really open to any suggestions and new ideas about initiatives that you'd like to see the network provide. So please feel free to email us or to reach out to us on Twitter with any ideas you may have. Ways to reach us will be put up into the chat box shortly, including a sign-up link if you're not yet a member, or if you'd like to receive newsletters, which you, if you don't already, and our Twitter profile page as well for you to follow on with upcoming events and discussion. A little bit of housekeeping for tonight. The event is being recorded, so just bear that in mind. We had many people request this, so it will be made available to registrants later on. If you can please remain muted while the speakers are on, and to have your camera off just for bandwidth issues, but please feel free to turn both back on when the Q&A is on and to become part of the discussion. We'll be monitoring questions in the chat box, so just pop them in there whenever they come to mind, and we'll have a 10-minute Q&A and discussion session at the conclusion of each talk. We are trying to keep this short and snappy, so Maddie will be helping out with a bell at the 10-minute mark of each talk, and also towards the conclusion of the discussion sessions, just so that you know that we're about to wrap up for each one. I realise it's a challenge to fit in the answers to these tricky questions within 10 minutes, both for the speakers and for the discussion, but we thought we'd, if anything, leave you with a desire for more information rather than less. So let's see what we can get through just in a short event tonight. So without further ado, I'll hand over to Hamdi, who will introduce our first speaker.
Good evening, everyone. I have the pleasure of introducing Professor Georgina Hold, who is a professor of gut microbiology at the St. George and Sutherland Clinical School in Western Australia. In New South Wales. Her passion is to understand interactions between resident microbes and their host, and how changes in the microbiota impact human health. She conducts clinically relevant microbiome research with a focus on inflammatory bowel disease. And we have Professor El Omar to thank for her import from the UK, where they worked together for several years. So with a PhD in molecular microbial ecology, Georgina is ideally placed for addressing tonight's opening talk, the who's who of the human microbiome, and we thank her sincerely for agreeing to speak on this topic tonight. Thank you.
Thanks, Hamdi. Can people hear me?
Yes.
Yep. Great. I was giving a talk to a nutrition society in the UK a couple of weeks ago, and I started chatting along, and then eventually someone said, "We can't hear you." So I always like to check in advance. So it's lovely to be here. I am in South Sydney. I'm not in Western Australia. I was in Queensland last week, but you know, it's really nice. And as Hamdi said, you know, I'm very passionate about microbes and how microbes cause disease. And you know, I started in this this amazing field back in the day, actually last century, which kind of makes me feel really old. But I was a microbial ecologist by trade, and I haven't changed. It's just that the whole journey has moved forward, and now I'm a microbiome expert. So I still do the same stuff as I did back then, but my name got changed just because we're moving on. So who's who of the human microbiome, an intro to key microbes. That was my remit, 10 minutes, off we go. Okay, so we're I'm a gut microbial ecologist particularly, and I always forget that there's also a large amount of the body outside of the gut that I need to remember when I'm talking in these kind of talks. So we are co-inhabited by a number of trillions of microbes. We have body habitat specific niches, and so the microbes tend to collect in these different habitats. They have to adapt hugely to those changing ecosystems, whether it just be through life or through the presence of different environmental factors or through the progression of disease, or and microbes really are helping to finely tune and responding to that changing physiology. So one of the things that I want to impress on you through this 10-minute talk is we're very focused on who's who in microbial ecology, but really we need to be thinking about what they're doing, not what their name is. And that's harder to do, and that's where we're certainly moving in the field. So we do have body-specific microbial consortium, but if you actually look, and I'll show you some data that really emphasizes this, it may be different microbes in different areas, but actually the functions that they're performing for us are actually broadly similar between ecosystems. So this word that's bandied around in terms of changes in microbial diversity that are associating with disease is dysbiosis. Now, dysbiosis is, you can't define it. It just means a change. It means a departure from the natural, balanced, healthy ecological state. We tend to associate dysbiosis when there's a perturbation or where there's a prolonged or an exacerbated change that is detrimental to health. But you can't say this change in microbiota defines dysbiosis, because it can mean a change in composition, but it can also mean a change in functional capacity as well. And in the context of inflammatory bowel disease, that can be a depletion of those key beneficial species, or it can be an exacerbation or an excess of those potentially pathogenic species. So if we have a larger an increase in proteobacteria or infuser bacteria or mucin degrading bacteria, these tend to be the not so good guys. This tends to correlate with a depletion or a loss of what we consider to be the good guys, the firmicutes, the butyrate producing species, the Faecalibacterium prausnitzii, the Roseburia species, the Ruminococcus species that are doing many of the good things. And so it's all about balance. And so when you talk about dysbiosis, you have to think about age, you have to think about geography, location, dietary status, as well as the disease that you're actually looking at, as well as the body site that you're also interested in. So this is some data. It's nearly 10 years old now, from Curtis Hutton-Howard's group, the Human Microbiome Project, but it still stands as a fantastic example of what I'm talking about. This was a paper that was published in June of 2012, and I remember it because I was actually due to give a talk on the healthy microbiome on the day after this was released. So there was a whole panel of papers published in Nature and Science back to back. I had a very busy night that night, understanding and being able to pull these into a talk that was already written. But what this data was, 242 healthy individuals from America, predominantly Caucasian, North Americans, looking at sampling the microbial diversity at various body sites. And these are the various body sites. And this is the bacterial composition defined broad classes as different colors. So you can see in the nose, in the anterior nares, you can see that we've got a lot of red, indicating there's a large number of actinobacteria in the nose. And so each column is like one individual. And so you've got multiple people showing you that spread of healthy individuals, where some people have a lot of red on the left-hand side, a lot of actinobacteria. Some people still healthy on the right-hand side of that anterior nares box have a large number of proteobacteria. And you can move through every body site. And if we focus then on the one second to the right, the stool sample, you can see that that is predominated by the blue and green. So some people have large amounts of Bacteroidetes, some people have large amounts of firmicutes. Everyone seems to have a wee bit of proteobacteria and maybe a little bit of actinobacteria or Tenericutes. But essentially all these people you would define as healthy. So what does a healthy microbial signature look like? we can't really say based on this information. So this variation shows you that the composition is not the answer to what is healthy. And moving more towards, if you look at this, if you translate the predicted functional capacity of these microbes, you see that it's much more stable across the different body sites. So again, it's saying that different microbes can contribute different metabolic capacities. And it's not species driven, and it's certainly not static. And so one thing that we have learned over the last decade is that we need to describe behavior of microbial communities over time if we want to get a good understanding of what's going on. So looking in the gut, we now know that there's over 1,000 bacterial species that have been classified, consistently dominated by Bacteroidetes and Firmicutes. And this knowledge has led to extensive reclassification. When I started back in this field, Bacteroides was generally a single phylum. Now it's split up into Aleostipes, Prevotella, Paraprevotella, Parabacteroides, and Odoribacter. What we find in Bacteroides is that we tend to have a lot of a limited number of bacterial species, whereas in Firmicutes, the taxonomy is more challenging. It's a more polyphyletic group, but we have a lesser number of more species. So there's challenges in understanding that taxonomy. And I just want to show this slightly out of focus. I'm sorry, I couldn't get a better picture of this, but this is just showing you the characterization of human stool in blue compared to mouse stool in red, just to show you that there are strong similarities, but also great differences in the microbial collections present in these ecosystems. So if you look at Bacteroides down at the bottom, you can see the red mouse species pretty much don't overlap with the human species. Whereas if you look in the Firmicutes, there's a lot more species available. You tend to have a little bit more overlap. So again, this is just showing you that in a completely different system that's evolved independently, we have a similar consortium, but they're not the same. And this sort of shows that mice are a good proxy, but they're also not the ideal proxy if we're wanting to look at what's going on in humans, because although they have similar species, they're not actually the same. So this is some more data, and again, it comes from the Huttenhauer group, and I don't apologize for that, because they've done an awful lot of the pioneering work in terms of understanding microbial changes in the context of disease. This is just really showing you in panel A, those main species, who's who in terms of human microbiome. So we have in the gut, we have the Bacteroidetes, we have the Clostridaceae, the Prevotellinaceae, the Eubacteria, the Ruminococcus, all of those fit into the Bacteroidetes and the Firmicutes. We also then have the Bifidobacteria and the Lactobacilli, and the Enterobacteria, Saccharomycetes as in fungal, and also Methanobacteria as in Archaeobacteria. So this is starting to bring in that there's other species, other kingdoms other than bacteria in the gut. And it's the same if you look across the skin, across the female reproductive tract, and also the mouth as well. And it's interesting that everybody will have different consortium, but they're all falling within that similar groupings. If we move to look at the functioning of these microbial groups, then we can see broadly across all body sites, we see this broad conservation of pathways in terms of carbon metabolism, cell cycle, structural protein synthesis. We see a drive towards the healthy microbes exhibiting colonization, resistance against pathogens. We also have the healthy microbiota educating the immune system through the presence of PAMPs, such as lipopolysaccharide and peptidoglycan, and also viral PAMPs as well. But then if you look, you have body site specific functions as well, which would make sense. In the mouth, we see a lot more involvement of pathways such as carbohydrate breakdown, regulation of pH. In the skin, we have the capacity for vitamin D synthesis. In the female reproductive tract, we have the regulation of pH, obviously via the lactic acid producing bacteria. And in the gut, we have a prevalence of bacteria that are producing short chain fatty acids through complex carbohydrate fermentation. Many of these bacteria belong to the Firmicutes, particularly the butyrate producing bacteria. The Bacteroidetes will also produce short chain fatty acids, but not butyrate, particularly more acetate and propionate. And so all of them are doing these fantastic sort of metabolic functions. And the last thing I really want to talk about is the fact that these microbial communities are changed by geography, by exposure to antibiotics, exposure to dietary components, colonization changes through genetics, and also diet as well. And it's very true to say they are the microbes, they are what we eat. And so, you know, we have a massive capacity to influence the microbes, but we have to understand what's going on before we can, in the healthy context, before we can start to make understandings in the disease context. Panel D is really showing you what drives my thought processes at the moment, and that's about whether a healthy microbiota has a stronger capacity to resist change, to resist perturbation. Is it a more resilient microbiome than that's present in a disease community? And so that's really something that we need to understand, that the microbiome is a collection of parts, and every part on its own plays a tiny role towards that kind of metabolic capability, but they're talking to each other and they're talking to the host, and it's that collection. We need to consider them a body, like an additional organ, which is what we've described them as before, rather than pulling apart all the individual pieces. And the last thing to say, I haven't really covered it too much until I came onto this slide, is we know an awful lot about the bacteria. We don't pay half as much attention to the fungal, the viral, or the protist and the eukaryotic protist and the archaebacteria as we should do. Why not? Because we don't understand them enough, we don't have enough information, and we don't know how to analyze them. It's getting better, but we still need to be better in these areas as well and understand their interaction with bacteria. So what defines a healthy microbial sort of functional capacity? It's one that can sustain life. It can enhance transcription, translation of those important factors. It's a microbiome that can harness energy production and to generate structural components that are necessary. It's able to communicate with the host. It can produce factors that can keep it there in kind of harmony, and factors that make it useful to be there so the host doesn't want to get rid of it. We have body site or niche specializations such as short chain fatty acid capacity or vitamin production. And I really like this quote that came from that same paper. A necessary condition of the healthy microbiome is the presence of, sorry, I've lost you there, is therefore the presence of an assemblage of microbial species that can carry out specific sets of biomolecular functions in each of the niche specific biochemical environments across the body. And that really, really helps you to understand what you need to know about who's who of the microbiome. So in summary, better understanding of the healthy microbiome and who's who is going to give us a better definition of disease microbial changes. We need to understand what the healthy boundaries of a microbial community are. And we really need to understand this in the context of personalized healthy medicine. We need to embrace that niche specific changes. So in a healthy gut microbiome, high microbial diversity, microbial richness is a really good thing. And so we think that we should apply that to all niches. No, not at all. High diversity in the female reproductive tract, for example, is an indicator of an unhealthy body site. We need to understand how microbial communities are able to resist perturbation, so looking at their resistance capacity, what their ability to return to a healthy state, whether there's a resilience in that microbial population. And we need to understand that we can't extrapolate on the basis of a single reference population, whether that be a single point in time or a single point in geography as well. So we have to extend our understanding. And then the last slide is just really what the current issues are. We have issues in terms of annotating the microbiota. These are limited because of our genetic information. So for example, 18% of the standard bog standard E. coli K12 genome remains unannotated, even though we have sequenced the hell out of it and we have complete closed genomes. So we still have 18% of the genome that we don't know what it does. If you look at some of the microbes that I'm interested in, more underexplored, such as Campylobacter shoei, over 50% of the genome is unannotated. So we know what's there, but we don't know what it does. And if you then multiply that by the hundreds and thousands of species in a microbial community, then you'll start to understand why we haven't got on top of things yet. We need more studies that compare health, and also looking at pre-symptomatic disease studies as well, because that's going to give us a better understanding of what the healthy components are that are going wrong. And we probably are only going to be able to achieve this by looking at kind of personalized monitoring approaches. And we need to integrate all available omics data sets, whether that be genomics, metabolomics, transcriptomics, single cell omics, culture omics, being able to actually isolate these organisms and have them to work with and to manipulate and understand what they're capable of is hugely important. I would say to you, if I took you and put you on a football field, and I put you in a freezer, and I put you in a desert, you are still the same person if I look at your genomics, but you will function completely different because your environment around you has changed. That's what we're asking to understand from microbes as well. So this is just to say thank you. This is where I work at the Microbiome Research Centre down at the University of New South Wales at St George Hospital. This is the cohort, this is our founding family of the microbiome researchers led by the director, Professor Imad El-Amar. And these are just some of the people that fund the research that the IBD team is running down at the MRC. And if anybody's interested, we'd be very happy to have anyone across to come and see what we're doing and what we're up to and what capacity we have. And we're very keen to and happy to collaborate. Thank you very much.
All right, fantastic. Thank you so much. We have a couple of questions that have been submitted in the chat. The first one is from Mark Reed, who asks, I see a lot of focus on bacteria in the literature. What proportion of bacteria of all microbes in terms of one, the quantity per cell, per organism, I guess, are actually present, and two, their impact on the host. So his example is, how much do we miss if we focus on bacteria alone?
Really good question. And as I tried to allude to, we tend to work with what we know, and we are very bad at talking about the microbiome and then only dealing with the bacterial component. I'm a very strong advocate for changing that, but I'm also guilty of expressing things in that way. We know that this interaction between the different microbial kingdoms is huge and very much under investigated at the moment. I mean, we've got kind of like we're starting to get a handle on fungal community diversity, and we're building much better databases of information. We're doing something similar in terms of the viral space. However, viruses are really complex. Unlike bacteria and fungi, where you can go in with a single housekeeping gene that will essentially be able to detect all bacteria or all fungi, viruses are not all the same. So we have doubles and single-stranded viruses, we have DNA, we have RNA viruses, and then we also have the bacteriophage that are obviously, their host is the bacteria. So it's really, really complex. But the more we look, the more we can find. And there was a fantastic paper in the IBD space a couple of years ago, back in 2014, looking at virome in the context of inflammatory bowel disease, paper published by a friend of mine, Skip Virgin, over from WashU University. And it was a really compelling paper looking at how Cauda viralis as a clade of viral viruses was hugely changed in the context of inflammatory bowel disease. And then about a year ago, the data was taken and interrogated in the novel, in newer techniques for analysing the microbial signature, and they found that while those findings were still relevant, if you actually mined and looked at the other viral components, there was a much bigger story there. So we're still constantly evolving the analysis approaches. One of the things we don't do we don't do very well or very consistently is actually look at numerical quantities of microbes in body spaces. It's very hard to do. And yeah, so when we're talking about relative abundance when we're really talking about the different microbes. And I think looking at absolute quantification and getting an idea whether numbers are dropping or increasing is also hugely important, but we just don't do that at the moment.
Thank you so much, Georgina. Just to wrap up, we've got time for one more question. And this one comes from Anthony Hanan, who wrote in the chat, do the gut microbiota of humans and mice differ because of their intrinsic/innate biology and or because of the standard chow/what we feed them in the lab that differs from humans? So how much of it is innate difference and how much of it is because of the laboratory conditions?
Again, it's a really great question, and I think you could speculate on every reason under the sun as to why there are differences. So we know if you change a human's diet, in 24 hours, their microbial composition of the gut will be hugely altered. So you just need 24 hours of switching from a high fat to a high carb or vegetarian to omnivorous diet. 24 hours is all you need for you not to be able to identify that person based on as what they were before, based on their fecal microbiota composition. We know that you can hold mice in different facilities and the microbial composition changes. Yes, just change their chow. I mean, we've been doing some work where we've been feeding mice high-fat diet. Yes, absolutely, it will change. But what we're really talking about is a community of microbes that seem to like to be together. And I think that there will be intrinsic host physiology factors differing between mice and humans as to why we change things. For example, mice don't Campylobacter won't colonize if you feed them to mice. So we can't, and you know, they'll quite happily colonize the human gut. So, you know, there's lots of very slight and subtle differences. But again, it's just to highlight that sometimes mice are not the best proxy for the human model, and there's things that you can't learn unless you do use the human model in whatever format you choose to use it. You can humanize the microflora, the microbiota of a mouse as well, and sometimes that can be a halfway house as well, but the mouse immune system wasn't developed to see those specific microbes, so who knows really.
Thank you.
Thank you very much for that. I'm going to chime in here because I think I heard a bell and we're just about on time. So things are looking great. Thank you very much for those questions. I see that there are a few more in the chat box, so I'm sure that we'll get an opportunity to post those and look at getting some answers for you at some stage. So with such a focus on function in that talk, I think that this has been a really nice segue into the next presentation, which I am delighted to announce. We have now got Associate Professor Andrew Holmes, who is a microbial ecologist at the University of Sydney's School of Life and Environmental Sciences. He's fascinated with the human microbiome and leads a research on diet microbiome interactions and how they relate to human health and disease. Now, he is a true fan of the natural world, and he's more likely to be watching birds than tweets, but he's certainly not camera shy, as you may have seen him on ABC's Catalyst as a resident microbiome specialist. Now, 16S versus shotgun metagenomics was really a topic, was one of the most popular, if not the most popular, in our pre-event tweet poll. So I'm very grateful to introduce Associate Professor Holmes, who will share his expertise on this topic tonight. Thank you very much.
Thank you, Hannah. I just tried to share my screen. I hope that's working and you can all hear me.
Absolutely, we can hear you and that looks great. Thank you very much.
You've given me a big and to some extent controversial topic to deal with in 10 minutes. So I'm not even going to try to go through the methodological details. What I thought I would do is take a little bit more of a conceptual view to this. What are the sorts of things that we might be answering with these different types of techniques? And I wanted to start with this rather funky looking image from Jerome Reyes and Pierre Bourque in a, or just over 10 years old review now. And what that illustrates is something that Georgina was just talking about, that what we're really interested in with the microbiome is working out how it actually functions as an ecosystem within the human body. And this figure here, I quite like that because it shows that right from the get-go, our field has been one that's technologically limited, and you can see a number of different things in here that have got little network connections between them that simply highlights that there's lots of room for complexity and different approaches in terms of how you might be trying to pull this system apart. So I'm going to go fairly simply here to take two people with their little microbiomes inside them. And I've got three questions there that you might be interested in. One of them is, does the microbiome of this person differ over time? That sort of question is going to be important if you're looking at the emergence of chronic diseases when your diet has changed or you've been exposed to an antibiotic treatment or even simply gone traveling. Another question might be, does the microbiome of these two people differ between each other? And obviously, if you're going to answer that question, you also have to take into account that either of them might also be varying over time. So what you're really asking is, when you look at the variation over time of this one person, is it moving around a centroid that's different to what you might see for this person over here? In other words, if they change their diet, are the differences introduced by their diet more or less than the differences you might see just between two individuals? And then finally, we might have these sorts of differences that we can observe either within one person over time or between people. What we're really more interested in again is, do any of those differences actually impact the human phenotypes? Now, this is where I want to get you to think a little bit, because from a clinical translation point of view, or even just simply understanding human biology, if we're going to get at this question, like, does this actually matter to our health and well-being or development? At some point, we need to actually identify and measure biological units in these microbiomes. And that's something that I think I could fairly safely say that, broadly speaking, the field simply has not thought about enough. Now, I've got a list of the sorts of things that you might be considering as units when you're wanting to compare one person to another. We've gone from the very, very pointy end, a 16S RNA molecule up here that you could collect and sample between these two environments and see if there's differences, all the way down to being able to categorize the whole microbiome as a community type, or this term enterotype has been bandied about, and say, is the enterotype of person A different to that of person B? Now, the point that I want to emphasize here is one that most of you have possibly not thought about too much before, but I hope you'll be thinking about it a lot after this. And that is that of this list of things here, only two of them are actually real physical biological entities that are unambiguously definable. The rest of them are conceptual units. And the two that are unambiguously definable are the 16S RNA molecule. If you have got one molecule of that, you know exactly what it is and no one's going to argue with you. Cell is also something where you can see one cell, although even here we start to see a bit of fuzziness go in, because if you've got a filamentous organism, do you count that as one cell or several cells stuck together in a chain? Everything below here is a conceptual unit that we've invented for the purpose of being able to categorize the system and then both compare one to the other, and also to be able to do maths, to be able to say, oh, I've got this many of this type in this system and fewer in this one there. If I multiply what their activity might be, I can say that their influence will be different. So how do we go about measuring these units? This is where we start to get at this question of the amplicon or 16S versus the shotgun sequencing. What I want you to think about, and I hope people will be firing questions in about this in the chat window, is that there's a few key topics or key questions that you need to ask yourself. The first is precision. If you are going to compare the 16S molecules or cells or genomes or species between this person and that person. You have to measure them in each person, and you have to be absolutely confident that the specimen that you collect from this person here, and then how you characterize it and assign it to being a particular type of 16S molecule or attribute that to a species, etc., must be reproducible and accurate. So there can be a range of sources of variation that come from how you actually collect the data, such as PCR bias, or how you actually categorize it and assign it to different operational taxonomic units, as you might do with 16S sequences, or to real taxonomic units, such as formally described species. Another issue that you need to think about is sampling effort or sampling depth. There's a mind-boggling number of microbes in the gut of each of these people, around about 10 to the 14. And if you only sample a few thousand or 10,000 or so of those cells from that person, and you try and compare that to another person, you can have error in your comparisons that's simply due to your sampling effort not being enough. So another question that you need to be asking is, what proportion of the total microbiome do your collected specimens represent? As Georgina mentioned, and I'll come back to again in a minute, there's also the informativeness or the resolution of what you're measuring. So if you measure these 16S molecules, it's possible for the cells of two different species that have different physiological properties to have an identical 16S RNA molecule. That's an example of where there's a limit to the resolution with which you can distinguish biological entities or cells or species that might be behaving differently within the gut. And that issue flows down all the way down this list as well. And then finally, we've got questions of practicality. How much time and resources are required to collect and analyse the specimens? So broadly speaking, we can split all of our different approaches into two: a taxon-centric approach or a gene-centric approach. And I've pulled out a little figure here from the same paper that Georgina mentioned earlier, but I'm focusing here on just the stool data. So in this image that you can see up here, essentially what we're looking at is a categorization of the microbiome of people by collecting 16S sequences and assigning them to higher bacterial taxa, which you can see listed over here. There's the phyla that were recognized back in 2012. And you can see immediately that at that level of resolution, there is enormous variation that you can see from person to person. If you were to repeat that sort of data on each individual over time, there's still variation over time, but it tends to not be anywhere near as extreme as what you see here between individuals. Below is something that was very interesting when it came out in this first paper here, and which in many respects sums up some of the differences between the gene-centric and the taxon-centric approach. There was a lot of interest in what the microbes might be doing, which you can't necessarily glean from this sort of analysis up here. And so with the advent of shotgun sequencing becoming possible, people were very excited at being able to look at genes that had been annotated with particular functions. And this data set that we're looking at here is at a coarse level of functional annotation of the genome, and it focuses only on those proportion of the genes that could actually be assigned. And what you see here is very, very little variation from one person to another. And this reflects that all bacterial genomes have got a common set of genes that are repeatedly observed to be involved in central metabolism and central housekeeping functions. So a gene-centric approach can still show differences from person to person, but it requires finer scales of annotation or functional attribution in order to be able to see those differences. So if we now start to get more to the pointy end of this, we've got our two broad conceptual approaches, the taxon-centric one or the gene-centric one. Now, I was asked to talk about amplicon sequencing versus shotgun sequencing, but what you'll see in this slide here is that actually the boundary is not so much about whether you're doing amplicons or shotgun. It's more about whether the sequence data that you collect, if you're matching that back to a reference database that's based on taxonomic characterization, or if you're matching the sequence data that you collect back to something that's about gene attribution rather than higher taxa that are based on cells and organisms. So the taxon-centric approach can be conducted with pretty much any sampling approach that allows you to make a determination of the taxonomic identity of the organism. So your specimens could be cells, and you see studies where people have looked at the microbiome by microscopy, and they've used various types of probes to identify the cells by microscopy or flow cytometry. Oops. You also see examples where the specimens are pure cultures, where they've recovered pure cultures and identified those cultures. Both of those tend to be relatively low resolution and take a lot of work to do them. Things that have really caused a revolution in the field are the sequencing-based approaches, and they can be either marker genes, such as the ribosomal RNA or the ITS region of fungi, that are amplified by PCR and then high-throughput sequencing of that, and then those sequence amplicons are matched to a taxonomic database. We can also do the same sort of thing with shotgun sequencing, but here, rather than matching one marker gene sequence, what we're attempting to do is to take any random sequence read that we find and assign that to what genome it may have come from. This is something that only in the last 2 or 3 years has become a more reliable or meaningful tool. And then finally, rather than using these just matching sequence reads to a pre-existing taxonomic scheme, we can de novo identify the species that might be present by genome reconstruction. So this is still using shotgun sequencing data, but the analytical approach is very, very different to try and reassemble the genomes. The contrasting approach is the gene-centric one, where we're simply collecting sequences and then assigning them to genes that we've got an inferred function for. That can be done in the reverse sort of approach, where you've done an amplicon sequencing analysis and used RNA sequences to come up with an identification, and then use a tool such as PyCrust, or there's a couple of other ones out there as well now, where you infer the genome and the distribution of sequence reads from the distribution of the taxa. Or you can directly do it from the shotgun sequence reads, where you're trying to attribute each individual read to a functional category of genes. Now I'm going to finish up with this last slide, which I hope will spark a little bit of discussion and a few questions from you, by summarising the different techniques that you might be applying, what sort of things can be measured with these, and a couple of quick comments that might pertain to that. Personally, I don't think that either RNA sequencing or shotgun sequencing is generically better than the other. I think that each of them have got applications to different sorts of questions, and that's the current policy of the ISME journal, where I'm one of the editors-in-chief, that we're quite happy to take papers that have used either approach. The key thing is that you use the most up-to-date reference database for your categorization or identification of the sequences. So culturing colonies has severe practicality limits of time and space, but you do get organisms that can be physically characterized in great detail and used for further experiments where they might be put back into animals to test what they're doing. The marker gene approach is where you have got the greatest capacity to sample in depth, cost effectively, and look at the dynamics of the community over time. So wherever you're wanting to look at how the community changes in response to some kind of environmental perturbation, this is going to be the most cost-effective way to do it. And now that we've got analytical approaches to infer sequence identity rather than using these 97% OTUs, this is becoming a very widespread approach. Shotgun sequencing is very useful if you're wanting to look for enrichment of particular functional attributes in one environment versus another. It's probably not quite so useful in the human context, because the environment of one human to the next is not as dramatically different as you might see in some other ecological analyses, but nonetheless it has its place. The metagenome assembled genomes is the one that's becoming the competitor with the marker gene approach for being able to look at community dynamics. And the real advantage here is that you can actually look at de novo identified species and have a little bit more information attached to them than you would if it was just a new RNA sequence that you'd come across. So I will leave it there with that very quick little talk and be prepared to be bombarded with questions.
Thank you very, very much. I think I speak on behalf of everyone when I say that that was extremely informative. It's a topic that everyone finds challenging. So thank you very much. So in the interest of time, we've got probably time for one quick question. However, I now see that the questions are flooding in. So again, we will get answers to those either outside of this session or in another method somehow. But we'll start with Lisa's question, which was in first, which was, do we currently have a good approach for dealing with the copy number variation problem in the 16S gene?
First of all, what I'll say is I'll attempt to answer all of the questions in the chat window while the next talk is going on. And with Lisa's question in specifically here, essentially it depends on the type of question you're answering. If you're looking at community dynamics, then you can largely ignore that question because the relative change is going to be proportional to the copy number variation. In other words, if species A had 4 copies and species B only had 1, that ratio of 4 to 1 will be preserved. And when you're looking over time, you'll still track differences there. If you're wanting to look for specific associations with a metabolic flux or something like that, that can become a little bit more of an issue there. But broadly speaking, I don't consider it to be an enormous problem except in specialized cases. Do you want me to go on to the next one?
No, thank you. We might just wrap up in the interest of time. I think you said that you might answer some of the questions in the chat box, so people keep on talking. I will introduce our final speaker for today, who is Dr. Thomas Quinn, in the Alfred Deakin Postdoctoral Fellow at Deakin's Applied Artificial Intelligence Institute. He is a medical graduate as well as a math and coding whiz. Best of all, Tom is passionate about applying his many skills to interpretable artificial intelligence as it relates to the microbiome and other omics. So he is a computational biologist we can all learn from, and tonight he will share with us his insights on computational data analysis, a topic that he's published on extensively. So thank you so much for your time, Dr. Quinn. Take it away.
Thanks for the warm introduction. Yeah, so this is a hard topic to really cram into 10 minutes, and I think I'm going to try to cover a bit of a breadth, and so that may involve me sort of gleaning over a few things rather quickly just to kind of explore this area and give you kind of a wide view of it. I think what I hope you get out of the talk at the very least is, as much as the microbiome is sort of like a new frontier in terms of natural science research and ecological research and human health research, it's also a new frontier in terms of methods as well. And, you know, part of understanding the data we collect involves methods. And I often get asked, you know, what's the best way to do it? And oftentimes there is no best way, or that best way is constantly evolving, or there are many competing best ways. So I'll talk to that a little bit, and mostly I think the talk is focused toward people who are actually sitting at the command line analyzing their data themselves, and hopefully you'll find that somewhat helpful if you've got data you need to analyze. So maybe I'll start, this is basically the outline, is to try to summarize what I think the problem is, which I would call, I would summarize as the question, why can't we just fit a linear model to the data that we have and call it a day? I'll talk a little bit about the different solutions to this problem and organize them into how I see it, sort of 3 prevailing schools of thought about how we ought to analyze microbiome data. I'll talk a bit about compositional data analysis specifically, transformations and references, and then try to end with a little bit of outlook if we have time. So what is the problem that we have? Well, I'm going to talk about transformations. What's the context of these transformations? Let's talk about differential abundance analysis as an example. This applies to microbiome modeling in general, with the exception of alpha diversity. It's really just a linear model in principle. Is you're trying to predict the abundance of an OTU or a gene or whatever your microbiome measurement of interest function as a function of some experimental condition or clinical condition adjusted for whatever confounders that you want. So why can't we just simply fit this linear model and then be done with it? Get our coefficients, report them, get the p-value, adjust them, and move on with our lives. Well, I think the answer largely boils down, although there are many minor answers. I think the major answer is there's this thing, a library size bias. So every sample is sequenced to a different depth, and that depth maybe doesn't really matter a whole lot in terms of if you have a deeper sequencing, that doesn't necessarily mean you have more bacteria in your gut, on your skin, or wherever. So how do you address this library size bias? Well, I think in my view, there are, generally speaking, three schools of thought. So I'm going to call them the absolutists, the proportionalists, and the compositionalists. So the absolutist sees the problems as the intersample biases. That's not too different from the other schools of thought, but they see the solution here as a normalization. So if we could know what that bias was, or what that bias factor was, we can just divide all their samples by that bias factor and reclaim the true absolute abundances of the data. So they may put forward as a null hypothesis, for example, that the normalized value of one of the OTUs or genes or whatever you're studying for group 1 is the same as the normalized value for that for group 2. The proportionalists see the problem as the arbitrary total, the fact that the sequencing depth varies sample to sample. And they would see the solution to this problem as rescaling the data to proportions. So everything sums up to 1, no more sequencing depth you need to worry about. And they might put forward a null hypothesis, something like the proportion of OTU1 for group 1 equals the proportion of OTU1 for group 2. Then you have the compositionalists, and the disclaimer is I sort of sit within this school of thought quite heavily. We see the problem as the constant sum constraint, which I'll explain a little bit later exactly what that means. But the solution here is to choose one of the OTUs or several of the OTUs, and again, that's genes or whatever you're studying, functions as well, as a reference against which to interpret the other variables that you have. And they would put forward as a null hypothesis, maybe something like the ratio of one of these bacteria when compared to some internal reference standard for one group is the same as the ratio for another group. So a bit of a Venn diagram of the different schools of thought. I don't think I'll have time to really go into detail, but the main point I want to highlight here is in red, which is that these three schools of thought can produce analyses that disagree with one another with respect to the linear models they build and the coefficients from those linear models and the p-values you get as well. And again, I sort of sit within that school, so I'm going to talk a little bit about this in more detail. So compositional data recognizes that the data are in fact proportions. And I think most of the microbiome world sort of sees this so-called relative data. Compositional data is a field that deals with relative data from before microbiome existed. All this data can be represented as pie charts or stacked bar charts if you prefer. And these proportions are quite special because proportions sum to a fixed total, so they all sum to 1. And because of that, there's this thing we call the constraint, which means that because everything must sum to 1, if one of the bacteria goes up, then it must by definition push all other bacteria down. And that makes it really complicated when you just analyze proportions of data by themselves, because did bacteria A go up or did bacteria B through Z go down? It can be impossible to know. So the compositional data people might argue. And because of that constraint, you might want to get some sort of reference that you can use as a yardstick for making sense of your data. Now, within compositional data analysis, which I'll talk a bit about because that's where I sit intellectually, there are many approaches to this, but I'll simplify it by saying there's basically one template that compositional data analysis uses, which you state some research hypothesis with respect to a reference that you're interested in. You use this reference to transform your data. You apply the statistics as you normally would, whatever that statistics are, say differential abundance analysis and formal linear models, but it could also be fancier things like clustering, regressions, multivariable analysis, multi-omics analysis, et cetera. You interpret your results with respect to that reference. And When it comes to choosing the reference, I think the people from the Coda camp would argue that there isn't really a right or wrong reference. Each one introduces a certain perspective. And I would encourage you as the domain experts, the expert microbiome people, to pick the reference that makes the most sense for the condition that you're studying. So what are the references that are available? There's all sorts of these things. I'll talk a little bit about the CLR because I think that's the most common one that's used, and I see it popping up more and more in the literature. But they all basically have the same form, which you take the log of the different variables you have, whether they're taxa-genes, et cetera, and you divide it by your reference. Now, all of these references have limitations, and I think that's something, I guess, the limited time to actually really talk about what all these limitations are in detail, but if there's nothing you get out of the talk other than this, is that references do have limitations. With regard to the CLR, which is the geometric mean, is that there's a lot of controversy, I think, emerging about whether or not the CLR is actually normalization and ought to count as a normalization. What does it actually mean when you CLR transform your data? So while it's popular, I would advise against using it without thinking critically about it. But a big issue really is that it's sensitive to the addition of removal or OTU. So when you do the CLR on your data, it standardizes it relative to the geometric mean of each sample. And that geometric mean changes depending on how you do the prevalence filtering, depending on the reference annotation database that you have. So that it can introduce some imperfections into your analysis. So as we're already getting, I think, 7 minutes or so in, we'll say that some advice, maybe if I were to just say like, all right, how would I advise if you don't really want to think that hard about this kind of analysis and you just want to say like, let's get some results coming in, how would you go about doing differential abundance analysis or your linear modeling? I'd say do some zero imputation, take care of those zeros somehow, because you can't take the log of 0. And there's basically you can just replace the zeros with some very small number randomly drawn from a distribution. Apply your CLR transformation, run your favorite linear model, and then bang, you can get your p-values, FDR, adjust them, you can write that paper out. But just make sure you acknowledge the limitations of CLR and think about what does that CLR actually mean when you're doing these per sample geometric means. And also think about how does the choice in zero imputation OTU filtering might impact your results. But I'd like to end with a little bit of an outlook and to say, you know, maybe we can flip the script a little bit. And instead of thinking so much just about modeling one OTU as a function of the condition, as we do with most traditional differential abundance analysis software, to think about, well, can we build models where we try to predict the condition as the function of the OTUs? And there should be plural on this second line here. It should be OTUs. So what combination of OTUs, what balance of OTUs actually is clinically relevant or ecologically relevant, whatever you're studying. If you're interested in that second line, well, we can formulate that as a machine learning problem and try to find some combination of OTUs and reference OTUs that gives you a nice condition, a nice predictive biomarker. And this is what we've tried to formulate as what we call the ratio biomarker framework. These ideas aren't all my own, but I've been trying to just clarify and make it easier to understand, easier to use. So we've got some genes up here or bacteria, whatever. We make some ratio score. So we're saying this is the familiar balance like Firmicutes to Bacteroides, like we saw some of the previous slides. So we've got this ratio biomarker, you're comparing these two things, and you're seeing how well do they actually predict the condition that you're trying to study. And then you use a machine learning algorithm to update this to find the ratio, find the balance that gives you the best separation between your class. And then you can go on and validate that in an external cohort, test that, et cetera. So there are packages already available that can do this in just a few lines of code. CellBal is the first one, and it's a really brilliant paper, and that inspired me to be involved in these second two packages, Amalgam and Codacore. And yeah, all right, I think that's pretty much 10 minutes. So final thing is I've got some papers here. If you're interested in reading, talk to me on Twitter, send me emails. I'm happy to talk. I'm trying to put some of this together on YouTube as well. I'm just happy to chat about these things. It's really hard, and I'm learning as well. And when you guys ask me questions, that's also how I learn more about this. So I'm happy to take questions now.
Thank you very, very much. A very challenging topic to get into 10 minutes again. So we appreciate how rapidly you got through it all and the breadth of content that you guided us through. Thank you very much for that. So we have a first question here from Mark again. it's a little bit long, so stay with me. A problem with relative abundance divided by total reads is that the total reads is arbitrary. It must all sum to 100%. One taxon goes up in relative abundance, another must go down, as you mentioned in your talk. For CLL transforms, doesn't the same still apply? I was of the impression that the data lie on a simplex. Apologies if I have misunderstood this.
Yeah. So I think, I mean, that's absolutely right. So when you do the CLR transformation, it's not a magical solution to the problem. I think it's been presented or at least interpreted by some as one. And I admit I made that mistake from probably 2016 to 2019. I wasn't thinking too critically about it. Now I'm sort of sounding the alarms and warning people not to make that same mistake. Yeah, the CLR is not necessarily getting rid of that so-called interdependence between the data. But what it is doing is it's putting everything into some center. It's centering the data, hence the center in the CLR. And that centering is sort of like a normalization. It's actually not technically very different to what DE-seq is doing and the Edger TMM implementations are doing. So maybe it's still useful in a way. Maybe the average of bacteria is a useful reference against which to compare all of the other bacteria to say, well, if there is differential abundance, if OTU1 goes up in this group, it's up relative to the average bacterial abundance. Maybe that's a meaningful concept. I'm not sure.
Yes, that I have to admit is slightly going over my head. So I'm going to have to definitely wait until your YouTube videos come out so I can upskill in that area. But no, I'm sure that the more mathematically aligned brains in the room are loving a lot of this. And I see some very, very happy comments coming through. Following on from my point that I am very much a novice in that space, do you have any recommendations on where, the average microbiome scientist could upskill in this area so that they were able to at least, apply some thought process behind it and be able to interpret things well or have a good conversation with their statistician?
Yeah, so I mean, this is going to be very self-serving, I suppose. But I've tried to write about this because this is sort of like my thing. This was really hard for me to understand as well. It's not easy stuff. I've just spent now like 6 years doing it, so I think I've got a bit of a handle, and I understand it thanks to people in the audience. Like Jonas was here for a little while. He's a real mathematician. He's taught me a whole heap. I've tried to write a review about why people think about it this way, and this is very much a conceptual thing. We've written a little field guide, which is about like, how do we actually operationalize this? How do you use it in practice? And then quite recently, I've become a bit more disillusioned with differential abundance analysis in general. And this is my argument why I think we ought not to do differential abundance analysis and instead be doing this kind of biomarker searching kind of thing, looking for balances that are clinically useful from the get-go. I hope they're approachable articles. I've tried to write them to be approachable, but the thing is that if you read it and you don't understand something, email me and I will try to explain it to you, because that's a failure on my part as a writer if you don't understand what's being said. The ideas here are not that hard. The math is actually relatively simple. It's just conceptually difficult, but biologists are good at conquering conceptual difficulties. So you can understand this. Excellent.
That gives me hope. I feel good. Well, you've got a fan. Someone says the field guide is great.
Oh, great. Yeah, I'm glad you ever. Yeah.
I'm just going to quickly take this opportunity to jump in, guys, just in the interest of and making sure everyone can get back and eat their dinner and put their kids to bed and everything. But I didn't want to interrupt that because it was great to see such in-depth and engaged conversation between everyone. So I very much encourage you guys to continue that activity online, get it on Twitter. Maddie has provided all of the relevant contact details and social media details for the network. So I very much encourage you guys to find us online, sign up and be a member if that's something you're interested in, or sign up for the newsletters so that you can stay on top of the activity of the group and also the various achievements and interests of the group members. And with that said, I'd like to really send my absolute heartfelt thank you to all of the speakers for tonight. I think it was incredible to see such a nice combination of speeches that were so truly complementary to one another. We started with this overview of the key players in the microbiome space, where functionality was a really important thread and emphasis of that presentation. And this was then complemented by some description of the various methods and their respective limitations on how we can look at functionality in a little bit more detail, and how we can approach this complex question of what technique to use for what question. And I think what we learned from that was that it depends on the question. So certainly an area to read into a little bit more for us all, I'm sure. And then it was wonderful to hear Tom finish up with the latest approaches on how to deal with this complex and very detailed data that we all work with on a hopefully not daily basis, because that might send you a little bit insane for many of us. But it is a challenging space that we're all in. We're potentially all on the forefront of this area. So it's going to be really exciting to see where this goes. And yeah, I am very appreciative of all of your time tonight, particularly the organisers as well. Maddie and Amy put a lot of work in behind the scenes. So thank you both very much for that. And thank you all for attending our first event. It was great to see so many numbers register and attend in real life. We call it real life now, I suppose. Zoom is real life, basically. And hopefully, one time in the near future, we can hold a real life event where we can network for longer with maybe a glass of wine in our hand. So, Amy, I'm not sure if you Amy or Maddie wanted to end with any quick last notes or practicalities.
Nothing else from me, just a big thank you and apologies again for the multiple Amy's online. You probably only have one. Good night, everyone. Thanks
you need
so
that
much.
in real life, Amy. Imagine that.
It's like being John Malkovich, being Amy Lockman.
Alrighty, let's sign off. Thanks so much.
Thanks, guys. Have
Thanks
a great
a lot.
night.
Well done.
Thanks for hosting. Bye-bye.
Thanks, everyone.